Good day. Have you ever wondered what the tiny, invisible world around us looks like up close? Today, we're going on an incredible journey into this hidden universe with the help of micrographs. Imagine seeing the fascinating shapes of bacteria that help or harm us, all blown up in stunning detail. These images not only satisfy our curiosity but also play a crucial role in science and medicine. We will uncover how to read and understand scale bars in micrographs. This is a continuation of our discussions on microorganisms. For more on the different types of microorganisms, please check out the link below. At the end of our video, we have a question for you to try out. You can test yourself with it before we show you the answer. It's a fun way to see how well you understand the topic. Practicing with these questions not only helps you grasp the material better but also boosts your confidence and performance when facing tests and exams. Otherwise, let's dive into the world of micrographs. A micrograph is an essential tool in biology and other scientific fields for studying microorganisms and structures that are not visible to the naked eye. Microorganisms are living organisms that are too small to see with the naked eye and which are only visible through a microscope. But what is a micrograph? A micrograph is a photograph or digital image taken through a microscope that shows a magnified view of a tiny object, such as a microorganism, cell, or tissue. So, a micrograph is a magnified image of a microorganism. A micrograph allows us to see what the microorganism looks like up close, but it makes it difficult to imagine how big the organism actually is. Since microorganisms are too small to be seen without magnification, micrographs are crucial for scientists to study and understand their structure, behavior, and function. Why are micrographs important? Firstly, micrographs allow scientists to see and study microorganisms, cells, and other tiny structures in great detail. Secondly, by using different types of microscopes, micrographs can reveal the details of cell components such as the nucleus, mitochondria, and ribosomes. Lastly, micrographs help in diagnosing diseases by allowing doctors to see bacteria, viruses, and changes in cell structure. How are micrographs created? Well, the specimen or sample is prepared and placed on a slide. The sample is magnified using the microscope's lenses. The magnified image is then captured using a camera attached to a microscope. Why is there a scale bar on a micrograph? In many cases, a scale bar is given with a micrograph. This scale can be used to calculate the actual size of the microorganism in the micrograph. The scale bar is a line or bar on a micrograph that represents a specific length in the actual specimen. It helps to understand the real size of the magnified object. For example, a scale bar might indicate that 1 micrometer in the micrograph corresponds to 1 micrometer in the real world. A micrometer, also known as a micron, is a unit of length in the metric system. It is used to measure very small sizes, especially those smaller than a millimeter. Micrometers are used to measure things that are too small to be seen with the naked eye, like cells, bacteria, and tiny particles. 1 micrometer is 1/millionth of a meter. 1 micrometer is equal to 1/1000th of a millimeter. There are 1,000 micrometers in a millimeter. Imagine a millimeter on a ruler. Now, divide that millimeter into 1,000 equal parts. Each of those tiny parts is 1 micrometer. So how do we use a scale bar to estimate size of a microorganism in a micrograph? A scale bar indicates by how much the image has been enlarged. For example, if you measure the bar with a ruler and get 8 mm and it's written 200 microns on the bar, then everything in the micrograph that is 8 mm long is 200 microns in real life. To get the actual length of an organism from a micrograph, we can follow these steps. Step 1. Measure the length of the scale bar. Step 2. Measure the length of the organism in the micrograph. Step 3. Divide the length of the organism by the length of the scale bar. Make sure you divide millimeters with millimeters only. After this, multiply your answer by the length printed on the scale bar. Step 4. Add the unit of the scale bar to your answer. The units on the scale bar are usually in micrometers. Let us move on and determine the actual size on the cocci bacteria on the image on the right. Step 1. Use your ruler to measure the length of the scale bar that has been provided with the micrograph. Measure the bar in millimeters. In this case, the scale bar measures 8 millimeters. This scale means that 8 millimeters on the photograph is equal to 200 microns in real life. Step 2. Use your ruler to measure the length of the bacterial cell in the micrograph. Make sure your measurement is in the same unit as your measurement of the scale bar. In this case, the cell measures 24 millimeters. Step 3. Calculate the actual length of the bacterial cell. The length of the bacterial cell in the image is divided by the length of the scale bar. The answer is then multiplied by the number on the scale bar. This means we divide 24 millimeters by 8 millimeters. The answer is then multiplied by 200 microns. If we do the calculation, we get an actual size of the bacteria of 600 microns. As we conclude, please attempt the following question before the answer pops up. You can pause the video as you go. In our next video, we will investigate the factors that affect the growth of yeast. Yeast ferments sugar to produce carbon dioxide and alcohol. You can check the link on this video below. Please do not forget to subscribe so that you do not miss our weekly uploads. Otherwise, thank you for watching and enjoy the rest of your day.